Okay, so I'll tell you about our work on modeling chromosomes. That's actually, can anybody guess what this picture is? Medical students told me it's a karyotype, but if you zoom in, these are socks. It's an excellent exhibit in London in the Wellcome Collection. You should absolutely see it. My favorite model of chromosomes. Anyway, so let me start by posing a problem. And the problem is that in many instances in biological situations, you have two elements. One can be an enhancer, another can be a promoter, or other elements that need to interact despite of being some distance apart along the genome. And our usual way of thinking about this is that they can interact somehow in 3D. There should be some sort of 3D interaction between them, some looping interaction. Now, in many cases, this looping interaction can be regulated by some sort of an insulator in between these two sites. And the problem is that, so when the insulator is bound, this interaction is not detected, or is less likely to happen. Now, the problem is that this insulator somehow needs to control things that are happening at a very different scale than the size of the insulator. And the problem is the problem of scales for me. So if I look at, say, a few hundred kb region, the size of the spatial size of this region is a few hundred nanometers. The length of DNA is a few microns. The size of an insulator is 3 nanometers. And I have no idea how a 3 nanometer sized particle, a protein, can regulate something happening hundreds of nanometers away. Electrostatics is screened. There are no interactions. There is no direct transport, arguably, in the nucleus, and so it's really a problem if you think about it. The question is, how can it work? And that's what I'll try to answer. So a tool that, again, everybody in the field is using, and I don't think this needs a detailed introduction, is a chromosome conformation capture method, which essentially gives you a population average, in most of the cases, population average map of pairwise contacts interactions. Again, cells are first, you add formaldehyde to cross-link, then you ligate genomic regions to each other, then you isolate ligation junctions and sequence them. And so each pair-end sequence gives you a pair of interactions present in at least one of the cells in the population. And it's usually averaged over millions of cells. So these are the maps that that have been produced since 2009, and they reveal a number of interesting features of the genome. And before going into these features of the genome, I would like again to explain what we're trying to do. So our approach to this data is that we're trying to model this data from first principles. And when I say model, let me explain what it is, because it's very different from models you've seen so far. So we're trying to build essentially physical polymer models of how genome is organized in different cells, building ensemble models of conformations. We're not building models based on the data. These are a priori models, or Occam's razor approach. So we use some prior biological knowledge, or in most of the cases, prior biological hypothesis, to fold this region, to fold the region of chromosome that we're interested in. So the way we model a chromosome, we say, okay, one monomer is going to contain 3 nucleosomes. So that's just a computational limitation. And so we can model a few megabases of DNA, a few megabases of the genome at this resolution. This gives us a few, we can do tens of thousands of monomers. So that's an underlying polymer. Then we add some model-specific constraints and interactions. We can say these monomers will interact with each other. They will attract each other. Those monomers may repel each other. So I impose some additional constraints or interactions. And in some cases, I try to make these interactions and constraints molecularly reasonable. So they're going to be local, in a sense. There are going to be no magic fields emitted by an insulator. In some cases, I don't do this. That depends on the problem we're trying to solve. So after these interactions are imposed, I let the system equilibrate. I essentially generate an ensemble of conformations subject to stochastic thermal fluctuations of this polymer. In this case, it was confined to be in a cylindrical shape because we were interested in the model of the mitotic chromosome. So I generate an ensemble of conformations that represent individual realizations of this chromosomal region in individual cells, and then I average over them by doing essentially a simulated high C experiment, if you wish. and this gives me a map of interactions, and then I compare in the simulations, and then I compare this map to a real map, and I decide whether this is a good agreement or not so good agreement, and then I go back, and I either go back to the parameters of the model that I can change and repeat the whole procedure, or if I feel that I explored the space of parameters sufficiently and I didn't find any agreement, I'm saying this model is inconsistent with the data, I need to move on. And in many cases, you can do these claims just by looking at sort of certain characteristics of the maps that you obtain. I'll show you some examples. So that's the overall approach. That's what we call a model. So coming back to the problem, coming back to Hi-C data first. So what are the most prominent features of Hi-C data that are of interest for us now? And sort of first of all, these are compartments. And again, I'm talking about mammalian Hi-C data, to be specific. In yeasts, many of these features are absent, or at least have not been detected. So in mammalian chromosomes, what you see are alternating compartments. You can infer them by eigenvector decomposition of these matrices, but to a good approximation, what you see is that there are alternating sort of this red and blue regions, and the red region that we would call compartment A would interact more with another red region, and another red region, and would be depleted in contacts, and you would see depletion of contacts between red and blue, or A and B. And a caricature picture of this is that active regions, A regions, also there is a good correlation between gene activity and identity of the region. So if one of them are, say, red ones are active, so then you see general enrichment of interactions between active regions, enrichment of interactions between inactive, and some depletion of interactions between them. That's a cartoon, so we don't really know what the mechanism of this is, how it works. That's still a mystery. What people have shown more recently, again, that became apparent in the 2009 Hi-C paper. So what became more clear more recently is, and these are two papers from Bin Rand's lab and Edith Hurd's lab, published in 2012, is that if you do the same analysis at a higher resolution, and you zoom into one of these compartments, you start seeing domains that are quite different. And they call them TADs, topological association domains. And that's a very nice example of an abuse of a mathematical term in biology, because they arguably have nothing to do with topology. But if you look at them, what makes them different from compartments is that there is no alternating, there is no checkerboard pattern. There are no alternating interactions. They might be nested, but they're not alternating. So what these TADs are, we also don't know, and that's the question we would like to answer. What also became apparent in this original paper by Edith Hurd and sort of the first author is Ilfaz Nora is that what's important for this domain information is something that sits at the boundary. Because when this element has been removed, and that was a 5C data, so when this element is removed, you see that the boundary shifts to the right and you get a new boundary. So kind of they propagate. So that was 2012, kind of. And there were many papers, more recent papers, that essentially found similar, kind of came to the same conclusion. That when domain removal, so boundary removal, inversion, mutation of CTCF sites, and so on, essentially lead to propagation of the boundary, to the next, to some other element in the genome. Another interesting paper that came out last year was a paper that showed that these changes in the boundaries are actually highly important from the functional point of view. And here what was shown is that a human phenotype, essentially a limb malformation, was found to be associated with the loss of a region in the boundary that contains a boundary element. And what was shown by the group of Stefan Mundlos is that removal of this element in mice leads to activation of these genes by an enhancer located in another domain. So essentially, enhancers start talking to these promoters if they're not properly insulated. So that's practically the cartoon picture that I showed you before, and then they've seen similar things with other domains, and they fully reproduce this phenotype in mice. Most recent paper, just a few months ago, have shown that a similar situation, to some extent similar, is observed in IDH-positive gliomas, where CTCF sites are methylated through the genome, including CTCF sites at this particular boundary. And again, it leads to the leakage of enhancer activation from this enhancer over to this gene, which is a prototypical glioma expressed gene. Overall, what it tells us is that boundaries control, elements at the boundaries control boundaries of TADs, and they also control interactions between elements contained into neighboring TADs. And these interactions are not only spatial, not only context, but these are functional interactions. And again, we're back to the same problem. How can something small at the domain boundary control interactions practically half a megabase away in each direction? And the idea that we have is that there might be a process, again, it's a complete hypothesis at this point. So there might be a process that can create such domains and boundaries, and when boundaries are removed, things would propagate. And the central idea of this mechanism is the concept of loop extrusion. And let me explain this. So essentially what's assumed is that there is a molecule, let's call it a loop extruder or a loop extruding factor, that can land on DNA or a chromatin fiber and can start progressively enlarge this loop. It doesn't bring things together as everybody believed like for last 10, 15 years. It lands on a piece of DNA and kind of enlarges a loop. And I can also illustrate this by this kind of arches. You'll see one of those arch diagrams later. This idea is borrowed from a study that kind of very systematically examined loop extrusion as a mechanism of chromosome condensation. That's a recent paper by Alipur and Marco, 2012, and where they said, what if such loop extruders exist and they can progressively enlarge loops? Can this compact the chromosome sufficiently well? And they studied a one-dimensional model, so there is no 3D model. It's just a one-dimensional model of this arch diagrams that can be created if these molecules create loops and then fall off, and I'll tell you more about this in a second. And again, it was even an earlier proposal that there might be this loop enlargement mechanism from Kim Nasmut, who in 2001 published a 72-page review. And in one paragraph says, oh, what if there is a loop enlargement mechanism? And that for mitotic condensation, because that would create loops and a scaffold of the chromosome. And he also suggests that that can also lead to segregation of sister chromatids. And again, I'll come back to this idea. So now what we're proposing is that the same mechanism might be operational during interphase. It might be mediated by different proteins. And in fact, we believe that in interphase this is cohesin, and in transition to metaphase that's going to be a family of condensing molecules. So what can cohesins do? So we essentially model this process following the steps of Alipur and Marco, but we're going to bring it to 3 dimensions. So you have cohesin landing, it creates a loop. It can fall off the DNA. If it falls off the DNA, the loop disappears. We also have, so if 2 cohesin molecules bump onto each other on this side, then they continue extruding loops on the other sides. You can think about this as 2 motors. 2 motors acting on DNA and pumping it. If it got stuck here, the other motor keeps pumping the material. So these are the assumptions of the Alipur-Marco model. And then we say, what if there is also a boundary element? And this boundary element, in our case, will correspond to a domain boundary. So what if at the domain boundary there is some boundary element, presumably a CTCF, we'll come back to this, that can stop propagation of this motor while the other motor will continue extruding. So that's essentially the model. So what we did, we took it to 3D. We say, let's simulate few megabases of DNA. Let's put these boundary elements. Let loop extruders exchange with the solvent. We're going to use our 3D simulation system. 1 monomer corresponding to 600 base pairs of DNA. It's all running on GPUs. The software was developed by Vijay Panda's lab. It's OpenMM, based on earlier incarnations of molecular dynamics. So we implement this constraints and active loop extrusion. So these are not only constraints. Loops are dynamically changing during simulations as protein jiggles. As DNA jiggles. So this just shows you one realization of the conformation of 3 domains, brown, green, and red. And then from this we can calculate contact maps. And then compare this to Hi-C. So the question is, what kind of features of these contact maps do I want to compare to Hi-C? So that's an example of very high resolution data from Erez Lieberman's lab, published in 2014, where they've observed beautiful TADs, domains, And also observe that some of them have, about half of them, have bright corners at the bright corners, they call them loops, kind of interactions between two boundary elements. So some of them do, some of them don't, but if I want to quantify this, I would like to look at the scaling of the contact probability, how intensity goes down within a domain, and that's going to be this green lane over here, and how the intensity goes down between two neighboring domains, and that's going to be the brown lane. So what I would love to get from my model, I would like to see whether it can quantitatively feed this contact probability plots, and also give me qualitative agreement with domains. So two very important. So the model in total has 6 parameters: DNA flexibility, DNA density, the rate at which loops are being extruded, and also 2 other parameters that are very important. Actually, one more parameter is the contact radius of high C. I don't know whether high C gives me contacts that are 50 nanometers apart or 250 nanometers apart, so that's another adjustable parameter. Actually, not adjust, that's another parameter in the model. And there are 2 other parameters that are very important. One is this linear separation between these loop extruders on DNA in kilobases, and the other is the processivity. Processivity is the size of a loop that a loop extruder would create if it were not obstructed by others and by boundaries. So it just lands on DNA, makes this loop, leaves. So that's the size of this loop. So when I scan through the 6-dimensional parameter space and select top 100 scoring models, they agree really well with the contact probability, scaling both between domains, between domains and within domains. I selected them by this criteria so I can find them in this 6-dimensional parameter space. And then it asks, where do they land on this diagram, this top scoring models? And they all land somewhere in this range of linear separation and processivity. So the size of each circle tells me how many models land there, and the darkness tells me how good is the fit. And so we're talking about 100 to 200 kb loops separated by, on the linear DNA, by 100 to 200 kb. Yes, Bill?
You said this and I missed it. What is the metric you're using to select the best parameter set?
Agreement on this thing.
On this,
Agreement on the scaling of contact probability with genomic distance. Let me slow down.
Is similar in different TADs, across different TADs?
So you see the variation is the width of this lane, and we did it separately for TADs of different sizes, because you cannot put on the same scale TADs of 150 kilobases and 600 kilobases. So we did it, we said let's focus on 360 kb, 300 range kb TADs. You can do it on smaller or larger ones. Any other questions about this? Okay.
This cohesion-like thing, it hops on and off randomly?
It hops. We assume that it hops on and off randomly from DNA. It's certainly not quite the case because we know that there are cohesion loading factors and cohesion unloading factors. But in the model, we assume, in the zero approximation, we assume that it lands on DNA and hops off anyway it wants. Just has a characteristic residence time. So it's a very simple model. I can add any sort of complexity on top of this.
So and this is, I mean, sorry.
Yeah, go ahead.
But this is known. So these things, they make a ring, and then this ring falls apart again, or?
It's known. What's known is that what's in the literature is that cohesions form a ring, and then there is a huge debate whether the ring connects 2 DNA pieces or whether each ring contains only 1 DNA piece. And that's what's in the literature since 2000. Actually, in the early 2000s, people spoke about cohesion being a molecular motor, and that was the time, near the time of Kim Nesmith review was kind of last mentioning of this, and since then it was completely forgotten. Since then, people have been talking about this kind of that cohesion just links things. So the notion of motors kind of disappeared from later reviews. Yes?
Is the loop extrusion and falling off happening on a different time scale from the polymer folding?
That's one of the parameters of the model. That's in fact, that's what I mentioned. The rate at which it extrudes relative to the time scale so it's a molecular time scale relative to the residence time scale. Okay, so as a result, what we see in the region that gives me good agreement is that we So the loop sizes are, as I told you, about 100 to 200 kb. On average, it's about half of the DNA that's in the loops, and another half is not. So it's not a huge compression of DNA. It's not a mitotic chromosome. It's a large interface chromosome, just maybe shorter linearly by a factor of 2, roughly. So qualitatively, that's how it looks. It's actually shockingly similar to what we see in Hi-C data. You see very nice domains. You see some of them having these corner peaks. Some of them having these flames. I should maybe show some examples with this kind of flames at the boundaries. There's many, many kind of visual features that we didn't actually quantify that resemble what you see in Hi-C. If you increase processivity, again, we don't know whether processivity is the same along the genome. Or the residence time the same along the genome. If I increase processivity, which is the same as saying that they reside twice longer, then I start seeing more of these corner peaks, and I start seeing some of the nested TADs. So there are lots of features that kind of immediately jump onto you in this very, very simple model. Yes?
Is this a snapshot, or?
It's a population average.
But what fixes these corners to be there?
It's a great question. So there are boundary elements here. So one cohesion hits at the boundary, and one side of a cohesion hits at the boundary. The other side keeps extruding a loop.
So those here are distributed initially in the simulation?
Boundary elements are set in the simulations. They're kind of fixed.
And you randomly drop them on the...
In this case, we just place them at specific positions. We've done also ask me at the end, and I'll show you a slide sort of at the very end of the presentation. So in this case, we just place boundaries and cohesions exchange. Yes?
You mentioned nested TADs. Could there be nested extrusions?
Yes, in fact, there are nested extrusions in this model because extruders can land anywhere in DNA, including other loops. This doesn't really change dynamics that much in this particular case because what's happening when the other one lands within a loop, it propagates until it reaches the base of the same loop, if the previous one was still present there. But because I'll come back to this when I talk about the dense regime. Okay.
How many elements are assumed to be there 100% of it? They always sit there, right?
In this case, they're assumed to be always there, but again, at the end, I can show you what happens if you make them kind of permeable. And they are now also bidirectional boundaries. So they prevent stuff from going in this direction, they prevent stuff from going in that direction. So it's it's an ideal gas model. I can add anything on top of that. So what does this tell me about the problem that I was trying to solve? And I think what it tells me is that, so in this picture, a TAD is not a loop. A TAD is a system of dynamic loops appearing and disappearing between two boundary elements. And the way it solves the so before going into this so it's a system of dynamic loops. Moreover, there are lots of spatial interactions between loops in neighboring TADs. It doesn't prevent spatial interactions. It just makes them less likely because you cannot have a loop that would involve a boundary element because nothing can propagate through the boundary element. But you can have spatial interactions between things on different sides of a TAD. Another interesting control is, I would say, what if I actually create one stable loop that would connect to boundary elements? And then I get a domain that looks very empty inside. And the scaling of the contact probability for this model would totally disagree with the data. So TADs are clearly not stable loops. They're dynamic loops appearing inside. There might be additional stable ones at the corners, and this stabilization actually happens naturally in the model. Another note is that it was shown in two papers. I should have cited them here. So that's the same paper by Aris Liberman that I cited earlier, and a paper by Susan Hajur and Amos Tanai's lab, where they've shown that CTCF at the boundary element, at the boundaries of TADs, has a tendency to be preferentially oriented. And what we did here, and in fact, when we look at what we did here actually looked at the top 4,000 CTCF peaks with CTCF motif such that we can orient this peak. And you see CTCF occupancy here in gray. And we also see a buildup of cohesin probed by these are two different elements of cohesin, RAD21 and SMC3. So you see a buildup of cohesin on one side of the peak. And we see exactly the same picture in simulations, that if that's my boundary element, I would see buildup of loop extrude on one side of it, because it's kind of knocking at the boundary element. That's a control. So what does this all tell me about how the problem of scales can be solved? And the issue is that the solution to this paradox in our mind is that that loop extrusion effectively replaces three-dimensional interactions, or supplements three-dimensional interactions with an effectively one-dimensional process of loop extrusion. And one-dimensional process can be insulated by a small protein, because this protein has no control over how these loops are interacting with each other, but it can prevent formation of this extruded loops. And if you look actually at the 3D picture of some of these domains, these are three neighboring domains, the pink, the green, and the blue. They interact a lot with each other. Nevertheless, they give me maps consistent with Hi-C maps in the frequency. So you need just to reduce this frequency by maybe two to threefold. And that's enough to get the agreement with what we see in the Hi-C data.
Is it enough to understand why an enhancer talks to a promoter in one case and not in the other?
It's a great question, and I don't have a definite answer to this question yet. So, with this, let me finish by showing what loop extrusion can do if I were to push it in a regime where I have many more loop extruders, and then there are going to be more landing within loops and kind of loops within loops. So what would happen if I now simulate the same process, and can it help me to resolve two problems that, again, Kim Nesmuth and John Marco were writing about. One is that if I have a chromosome, the chromosome needs to condense into an elongated shape, and the shape should be ordered. So if I paint this polymer in a rainbow fashion, red regions should be here, orange there, and so on. Whereas if I just condense this chromosome, it will be condensed into a blob with mixed colors. So how can I get this? The second problem is, can I disentangle two sister chromatids? Can I segregate into two separate chromosomes? And if I apply loop extrusion to the system, so these are simulations where we just take one polymer, and these are loop extruders, and they'll be exchanging and forming loops. These are going to be loops formed, shown as an arch diagram. The number here shows that this loop will be reinforced. You see, that's a loop within a loop then. And then the moment it reaches the big one, it becomes number 2, number 3, and so on. So these are loops that effectively get reinforced by multiple loop extruders, condensants, we believe in this case. What you see happening automatically is that there is a scaffold of a chromosome, and you start getting the right or shape of the chromosome and linear order of the chromosome. You get it all from a single process. There are no boundaries anymore, no additional interactions other than pumping of the loops. So that's in our mind resolves the first paradox, and then the second one is having two sister chromatids. And so I'll just show you this few second simulation of two sister chromatids. Again, you see extrusion of loops in each chromatid. You see that the formation of loops is kind of not perfect, so some of them kind of appear and disappear. As this is happening, as they condense, they start repelling each other just by excluded volume interactions, and they also shorten, become very stiff, and they disentangle. Again, this is happening by simple loop extrusion process. Again, as was hypothesized by Marco and Nesmuth. So with this, that's an argument why cohesins can potentially do this. Very simple graphical summary that loop extrusion might be a universal mechanism that is acting in the interface in a more dilute regime with boundaries, and in metaphase, more dense regime, so I need to remove boundaries and increase, presumably replace cohesins with condensants, and need to have many more of them, factor of 5 to 10 more of them. And that's a summary. We put this on the bioRxiv. These are the 3 papers on the bioRxiv. And these are people who did the work. Max did all the simulations for the loop extrusion. Jeff did all the data analysis of domains. Nizar analyzed ChIP-seq data, CTCF. They're all, so Jeff, Max, and Nizar are here. Anton is here as well. He did all the transition to G2 simulations and the theory of loop extrusion. And Carolyn Liu, she was a high school student when she started working with us. She actually initiated this project on loop extrusion as a mechanism for domain formation. She's now a junior at MIT, yes. Again, software for Vijay Pandey, and that's how these guys look now. Thank you for attention. Okay, questions? Yes.
So the CADs, so your idea that CADs very actively and inactively made and established, correct? So all the machinery all the genes that are getting turned on and stuff, I guess I'm not getting what's happening. I mean, there's a lot of machinery that's on that loop doing things. And do you think the loop's coming and going, or are you talking about little loops within it?
Okay, so there are I see two questions in your one question. One is, how do we see dynamics of loops? So we see that there are many loops appearing and disappearing. Increased frequency of contacts between two boundary elements might be a result of transient large loops, or maybe two loops that just brought these two elements closer to each other. In the data, we see that the intensity of interactions at the corner of the TAD is not nearly as intense as, say, interactions 5 kb apart. This is more like 50 kb apart. So it's clearly not a stable loop. And then the other part of your question, I think, is that how can I imagine this loop extrusion propagate through nucleosomes and all the stuff on DNA? And my answer is, I don't know. I have no idea. I can think of mechanisms how motors can hop over obstacles, and this is happening now. Myosins, and that's how myosins and kinesins move on microtubules and actin as we speak and move. So their step size is about 8 nanometers of kinesin. So it can hop over stuff on microtubules like without really seeing them. So maybe the same stepping model applies to cohesin. Again, in our mind, cohesin is a molecular motor acting on chromatin. How it works, maybe in another 10 years we'll know. Yes, Ben?
So what roles are cohesin playing versus condensin? What's the difference?
Cohesin is present in the nucleus during interphase. There is a cohesin present in G1 and in G2. In G2, it has another function of linking two sister chromatids together. The model is that cohesin forms a ring, and you have two sister chromatids. In G1, cohesin exchanges faster, and what it does, nobody knows. It's present. Its residence time has been measured.
But in terms of your model, and in terms of loop formation, they're essentially equivalent. They just have different.
They're just equivalent, even the same processivity, we assume. So, and condensin 2 is present during interphase, and condensin 1 comes in later. There is some controversy of what they do specifically, but in terms of model, they're practically identical. Maybe up to small adjustment of parameters, less than a factor of 2.
Why do you think that the loops disappear? Couldn't you get more compactification if you had actually nested loops and that it didn't?
Oh, no. Nested loops are fine. Actually, nested loops means that's what's happening with condensin. Again, it's nothing special about it. So just if I have more of them, then when I create one loop, it's very likely that somebody will land here. I have two neighbors on both sides. I have nowhere to go. And somebody that comes in will essentially form a loop, essentially reinforce the same loop. So loops become much more stable, because they're stabilized by a stack of condensers. Yes, another question, 2 questions.
The biology of condensin and cohesin is different in the sense that when you mutate cohesin subunits, you get separation of the sisters. So you're assuming, all the models that Naismith and Koshland have proposed actually think about the circle that holds the two sisters together. And you are talking about the circle that holds the same sister actually going up and down, right?
I don't know whether it's a single circle or maybe 2 circles, but yes, I'm talking about G1 cohesin.
Right?
Yes.
Okay, and that's one comment that I would like to make. And the other one is, there is a precedent in E. coli, in recombination in E. coli. You have pumping machines, helicases.
Exactly. Yes.
That actually pump the DNA until they encounter a region in the DNA called chi, which is a 10 base per recognition site, and then they stop. And so something similar could be happening here, in which you would recognize the CTCF binding sites or some protein sitting there or something like that.
That's exactly our thinking about this, is that, in fact, if you look at these two domains of cohesin, they're homologous to those helicases. that can move at a speed of 5 kilobases per second in E. coli. I don't think this moves at 5 kilobases per second because it has nucleosomes, but generally I agree that this might be a very similar mechanism.
We're going to have to stop now and thank Leonid again.